Hi, my name is Emily Perbula, and this is my mini exam video number 5. A protein that is 4 amino acids long is formed in vivo from this DNA coding strand. The first step in this process is transcribing the DNA coding strand to an mRNA coding strand. In order to do this, we must identify the coding and template strands. If we flip the bottom strand so that it reads 5' to 3', we can see both strands have the ATG start codon. To find the coding strand, we look for a stop codon that is 4 codons away from the ATG start codon. The bottom strand contains a stop codon, UAA, so therefore the bottom strand is the coding strand and the top strand is the template strand. Now we take the coding strand and transcribe it to mRNA by changing the thymines to uracils. We can now identify the amino acid sequence starting with the start codon AUG, which codes for methionine. GUC then codes for valine, AGC codes for serine, and AAG codes for lysine, followed by the stop codon. The first mutation I will describe is a Missense mutation. In this mutation, a guanine from the DNA coding strand becomes a cytosine. This change in the amino acid sequence would be a transversion, as AAC codes for asparagine and not lysine as in the original DNA strand. The second mutation is a nonsense mutation. In this mutation, an adenine is changed to a thymine in the original DNA coding strand. Then, in the mRNA strand, the new thymine becomes a uracil, as this is what happens in mRNA. The new codon, UAG, codes for a stop codon instead of AAG in the original, while that one codes for lysine. This example is also a transversion, as the adenine is changed to a thymine. The third mutation is called a silent mutation, and it is also a transition. In this mutation, the guanine is changed to an adenine in the DNA coding strand. This kind of mutation does not affect the resulting amino acid sequence. In this example, both AAA and AAG code for lysine. The final type of mutation is called a read-through mutation. In this example, there is a transversion in the stop codon. The TAA in the DNA coding strand becomes TTA as the adenine is changed to a thymine. As before, the TTA will become UUA as the original DNA coding strand becomes mRNA. The thymines are changed to uracils, and this will result in a change in the amino acid sequence as the stop codon has been changed. Therefore, the mRNA will continue being transcribed until the next codon appears.